Continuing here with the oligodynamic action of heavy metals. And we would have done an experiment on that in a lab using silver nitrate solution and copper sulfate solution. It works actually great. And another good thing about metal ions, besides them needed only in small amounts, is that bacteria will never be resistant to them. It's not like antibiotics where they get resistant to. These metal ions inhibit protein synthesis, denature proteins, so they are a really great way of stopping growth of bacteria. And here's some applications for you. Silver nitrate as an application would be these are the drops, the eye drops that newborn babies are getting. Mercury chloride prevents mildew and paint. Now, we don't really want to have mercury in our paint, but that's another story. Copper sulfate is an aldehyde. That means it reduces the growth of algae cells, but it's just as good for bacteria. And if you take a look in the lab folder for week 7, there are some videos posted for you that show you how hospitals are applying their knowledge that we have about copper and its growth inhibition of bacteria. So they're applying copper on hospital rails and beds. City of Hope, they were lining their duct system with copper because it reduces microbial growth. And then another thing you may have heard about, zinc chloride, is found in mouthwash. That's pretty popular. Here's an image that shows you something similar to what we would have done in a lab. We would have done the filter paper disc method, dipping our filter paper disc into a copper sulfate solution and then putting them on a lawn of bacteria and see what happens. But here what you have is, I guess this is some sort of a pendant, maybe a silver containing item, and you can see how it inhibits the growth of the bacteria right there. Pennies, copper pennies have the same effect, so oligodynamic action is a good thing. Now moving on to surface active agents. The most popular one definitely is soap, and soap reduces the number of bacteria. It emulsifies any kind of oily or fatty substances, and degerming means it just removes amounts of bacteria. It's not sterilizing your hands if you're washing hands with soap, but it's definitely reducing the numbers. Then there are acid anionic sanitizers, where the anions react with the plasma membrane. That's commonly not so common in households. That would be more something in hospital settings. And then very popular also in hospital settings are quaternary ammonium compounds. That's what you also might find in doctors' offices. Those are cations that are bactericidal, and denature proteins disrupt plasma membranes. So here you see an ammonium ion, and then here would be a quaternary ammonium compound like benzalkonium chloride that you can use as a disinfectant. There's a comparison chart of the effectiveness of various antiseptics. The steeper slope down, the better, of course. And so here, take a look at 1% iodine and 70% ethanol. So tincture of iodine, pretty effective in removing bacteria. And tincture of zephyrin, not quite as effective, but still pretty good. Aqueous zephyrin diluted 1 to 1,000, not so great. Soap and water looks pretty bad here, but it's actually not as well. It doesn't kill bacteria, but it's really effective in reducing numbers. Here's a clinical focus question for you. So think about it. Infection following anesthesia injection. Which preparation is more effective? Well, here are the two options. Undiluted versus 1 to 10 diluted for whatever antimicrobial substance that is. And you can tell on a disc diffusion test that the undiluted version of this antimicrobial agent is pretty effective. The diluted version is basically not effective at all. So we're going to have to go with the undiluted to be effective. Now moving on to chemical food preservatives. So how do we keep our food longer? There are different ways you can go. Chemical food preservatives include sulfur dioxide, that's added a lot of times to wine and prevents wine spoilage. Organic acids can be used, sorbic acid, benzoic acid, they're all very popular as preservatives for your food. You can take a look at bread packages. A lot of times they contain benzoic acid or sorbic acid. Those are very popular. These substances, they tend to inhibit metabolism of bacteria. They tend to be like false food that causes the bacteria to not be able to grow when they take that in. They think it's food, the sorbic acid or benzoic acid, and then it inhibits their metabolism. And that's how you keep your food items longer. Nitrites and nitrates, they prevent endospore germination. Nitrites are actually fairly often used as preservatives. Take a look at packages that have preserved meat products like beef jerky. They a lot of times contain nitrites. And now, of course, antibiotics. So we're going to have a whole chapter on antibiotics. So, and as far as using it for preventing food spoilage, of course, there you can add antibiotic substances. So there are some bactericidins, proteins produced by one bacterium that inhibit another. And we'll talk more about those in that antibiotic lecture. Niacin, natamycin, they prevent spoilage of cheeses. They're pretty often used to keep your cheese longer. And then aldehydes, they are very strong chemicals. You probably have heard of formaldehyde. If you've taken anatomy, a lot of the specimen are preserved in formaldehyde or glutaraldehyde. Those are not good chemicals. A lot of them are causing cancer, and they're pretty harsh chemicals. So they inactivate proteins by cross-linking with functional groups like the amino group here, or the hydroxyl group, carboxylic acid group, also hydro group right here. So they're very harsh chemicals, as I said, but they work great, of course. Now here is chemical sterilization. So there you would use gas molecules that cause alkylation, that works by replacing hydrogen atoms of a chemical group with a free radical, and that destroys cells. They can also cross-link nucleic acids and proteins. That, of course, will destroy cells. And ethylene oxide, that's a gas that's a lot of times used for heat-sensitive materials. So this gas works great. Now, this method you may not have heard about. There is something called plasma, and that's considered a fourth state of matter. So besides something being solid, liquid, or gas, there is this plasma state that consists of electrically excited gas, and it's free radicals that are highly reactive and destroy microbial cells. That kind of method is often used for tubular instruments. Then another method that you may not have heard about are supercritical fluids. So there'll be a CO2, carbon dioxide, with gaseous and liquid properties that can be used for medical implants to sterilize those. And then very effective in sterilizing something would be peroxygens and other forms of oxygen that are highly reactive. So those would be oxidizing agents. And peroxides like this here, peroxide H2O2, here O3, that's ozone. These are all considered toxic forms of oxygen, and they can be used therefore to destroy cells. And here finally, this lecture should have given you a little bit of an idea of what type of antimicrobial substances are out there. And something to always consider is what antimicrobial you might want to use, because the type of microbe that is affected by the different types of agents is different. So you can't use the same antimicrobial substance for everything. There are better and not so good choices for each antimicrobial agent and for each microbe as well. So here is a chart that shows you the effectiveness of chemical and antimicrobials against different types of bacteria. And ones that are very particularly bad to get rid of would be those bacteria that have endospores, and they are survivors. And mycobacteria, they are also very tough to get rid of. So here are some chemical agents listed and what they would be good for. Now, if you have an organism that has an endospore, for example, you might wanna, what can you use? None of these are effective, these are poor, and these are fair. So your best choice is maybe chlorine or glutaraldehyde, but none of it is really good. So getting rid of spores is really tough. Probably your best option is to autoclave it if you can do that with the material. Mycobacteria, same thing. They're hard to get rid of. None of these items would work. This one's fair. These here are pretty good. So phenolics, iodines, alcohols, glutaraldehyde. So those would be options. But the point here is that not everything works the same for every bacterium, and you got to really think, what do I want to get rid of? What type of bacterium am I dealing with most likely, and what substance should I use therefore?